Hello, honors biology, and today we'll talk about active transport. And under active transport, we have endocytosis and exocytosis. All right, so if you remember, again, the structure of the cell membrane is semi-permeable, and it has all the macromolecules we studied, the protein, bilayer of phospholipid, carbohydrate groups attached to protein or phospholipids, and cholesterol. Okay, and we'll go down to active transport for this screencast. So if you remember, we can categorize transport into two categories: passive and active. Passive transport includes transport that does not use energy, and it goes along the gradient. And if you remember, along the gradient means from high concentration to low concentration. And so under passive we have simple diffusion. Oops. Simple diffusion and facilitated diffusion. So under both of these is what we call passive transport. And passive transport again uses no energy and goes along the gradient. And again, what type of things simply diffuse? Things that are small. Oops. Small and nonpolar. And things that are small and nonpolar are things like oxygen, CO2, and your lipids. Okay, and facilitated diffusion allows things that are large and charged. Things like water, sugar, certain ions, okay, large and charged. And facilitated diffusion uses helpers like proteins that act like protein channels or gate. So both of these are under the category of passive transport. Now, under active transport, you are going against the gradient. And when I say against the gradient, I mean from low concentration to high concentration. So think of it like if you're going up the hill, right? You need to input energy and you're going from low to high. So you're going against the gradient. And to go against a gradient, you need energy. And that's usually in form of ATP. And what kind of things use active transport? Things like ions, like sodium, potassium, and hydrogen. Now, nerve cells are an example where they need to move ions really fast and against a gradient. And here's where active transport is used. And there's a special protein called sodium-potassium pump. And a sodium-potassium pump does is it pumps that sodium out of the cell, so you have a high concentration of sodium outside the cell and high concentration of potassium in the cell, forming a electrical chemical gradient. And how it does this is that by ATP attaching to this gradient causes a change in shape and moves 3 sodium ions out the cell and 2 potassium cells in the cell. So if you look at this next screen, it has a better picture here. So what happens is the 3 sodium ions attached to the protein, and when ATP here phosphorylates this protein, it changes its shape, causes a change in shape. And this change in shape then causes that sodium to be released out the cell. And then when you dephosphorylate, The word dephosphorylate means we remove this potassium. It causes a change in shape again. So the triangle is just a short form of writing change. And when the sodium here, 3 sodium, are now out the cell, then 2 potassium ions attach to the protein, and when it dephosphorylates, it changes shape and releases the potassium in the cell. So this is how we have sodium-potassium pump uses energy to pump sodium out and potassium in the cell. Another kind of proton pump is the hydrogen pump. The hydrogen pump pumps hydrogen, again from low to high, and we're going to see this in cell respiration, photosynthesis, and lysosome. And the whole purpose is, again, for this pump is to pump hydrogen, forming a concentration gradient of hydrogen in one side of the membrane to another side. Now, active transport also uses vesicles, and if you remember vesicles as a bag of cell membrane, and what happens is there's 2 types: endocytosis. The word endo means in, cytosis means cell. Exocytosis means out the cell. So if you look at this animation here, the cell membrane pinches in, and then you have a vesicle going into the cell. Where exocytosis is the opposite, where this vesicle will fuse with the cell membrane and then release the contents out. So there's 2 types of endocytosis. One is called pinocytosis. Basically, you could think of it as cell drinking or taking in small molecules, or when it's large particles, it's called phagocytosis. So it's basically taking the substance surrounding the cell membrane around it and then pinching off from the cell membrane and bringing it into the cell. And what this does is separates that particle from the different parts of the cell. So this could be taking that food particles and then taking it to the lysosome to get digested. So if you look at this picture, here's an amoeba engulfing a paramecium. So it surrounds its cell membrane around this paramecium and then engulfs it. And that's endocytosis. Here's another example of white blood cell engulfing a bacteria. And exocytosis is the opposite, where that vesicle will merge with the cell membrane and then release the contents out. Another example here is how the ER, the rough ER, and the Golgi body, again, work through a series of vesicles going out the ER and then into the Golgi body by fusing the vesicle in and out. Here's an example of insulin being released by pancreas cells by exocytosis. Okay. So again, active transport, you have active transport either using vesicles, or you have active transport using pumps like sodium, potassium, or hydrogen pumps.